So good afternoon, I am Dr. Murugan. Good afternoon, I am Dr. Murugan. I am going to teach biology. Basically, I am a professor in microbiology, teaching microbiology for UG, PG and research scholars more than 20 years. And very recently, I take up the job of teaching school students, and I started school teaching as in a competitive exam like NEET in Venpur as a part-time faculty teaching biology. Then joined first bench Chennai for the 2 months. Now I am taking home tuition for the 9th to 12th students. So right now I am going to teach genetics. In biology, genetics. So genetics is the study of gene. It's a science dealing with genes. So genes are made up of DNA as well as RNA. So this is DNA is known as central dogma because all the genetic information are reside in the DNA as in the form of molecules known as biomolecules. So the DNA is one of the biomolecules. Apart from that, various other biomolecules are there. RNA is a biomolecule, protein is a biomolecule, Golgi complex, cytoplasm, mRNA, tRNA and other things. Almost all the things which is found in a cell is known as biomolecules. But when comparing other biomolecules, DNA is very, very important because all the genetic information are resided in DNA. So this is very much important for the heredity. So heredity is nothing but how the characters are transferred from parents to offspring. So parents to offspring. Offspring means their progeny. So characters means the skin character, hair character, nail character, then their way of behavior, their metabolic pathway, everything coming under this heredity. So this hereditary information are resided in the DNA. Okay, so Indian DNA contains various other chemicals, that is it contains of nitrogenous bases, then phosphoric acid plus carbohydrate molecules. Carbohydrate in case of DNA is deoxyribose. It is a sugar molecules or sugar moiety. Okay, so all these things is a nucleotide. So the polymer of nucleotide is comprising this DNA molecules. So these DNA molecules act as a hereditary or the information stored in the DNA. In lower form, it is in the form of chromosome, single chromosome. Suppose in the bacteria, so bacteria have a single chromosome. So here the genetic material information is resided. Whereas in the multicellular organism, where it is in the plants, animals, human being. So this is a higher version of the biological things. Here it is in the form of chromosome, but it contains multi number of chromosomes, that is more than a single chromosome. So usually human being contains 22 plus 6 chromosome, X or XY chromosomes. So 23 pairs of chromosomes. Okay, so these all the 23 pairs of chromosome are taking information of the parents to the progeny. Suppose the parents have eye color of black, then ultimately suppose the male have eye color of blue and the female having eye color of brown, then ultimately the offspring from these two parents, the forming offsprings must contain either blue color or brown color. In some cases mixing of these two colors happens because of the genetic information present in the DNA. Okay, so the father of genetics is John Gregor Mendel, who is a monk, Austrian monk. Father is John Gregor Mendel, who lived in the year 22 to 1884. So he is an Austrian monk. Actually, his passion is to become a teacher, but he is unable to complete the certificate. Certificate for teaching. Hence, he denied of teaching, but he invented the things now we are teaching in the classroom. That is, he worked on the pea plant, and for his experiment, he took two kinds of plants. One is tall plant and having the genetic makeup of TT, and another is dwarf plant. It is generated as small t. In the first generation, F1 generation, all the plants, all the F1 generation are tall. Even though the genetic makeup is made up of capital T as well as small t, but all are tall. But in the pure breeding of the F2 generation, so he obtained 3:1 ratio of tall as well as dwarf. Okay, so this can be explained by means of checkerboard. Checkerboard is the board which explain the phenotypic character as well as genotypic character of a plant or the animal. So this is another side. This is one is male and another is female, you can imagine. So suppose this is a male and this is a female. So here the gene is formed by means of capital T as well as small t. Here capital T as well as small t. Sorry. So capital T, here small t, and finally here it is completely. So now, so this is TT, the organism which contains TT is a tall, and capital T as well as small t also tall, and so small t is a dwarf plants, and hence he obtained the tall plus as well as dwarf plant in the 3:1 ratio. So the genetic makeup is like, because this T is a dominant gene and T is a recessive gene. So if a single dominant gene is there, then ultimately the plants become tall. If two kinds of dwarf gene, then only it is able to express its dwarfness. So this is his first experiment. Based on this, he explained the character or resided in the gene which is found in the plant. And later on, so many people are worked on that, and in the recent advancement, so there's a bacterial genetics that leads to the biotechnology. Then human genetics, so that leads to the gene therapy. Gene therapy as well as human genome project. Okay, so then now we discuss what is bacterial genetics. So the father of bacterial genetics is Griffith. He conducted experiment in the bacteria. He took Streptococcus pneumoniae, so this is the organism which cause pneumonitis, that is lung infection in human being. He took two kinds of Streptococcus pneumoniae, that is, one is smooth or S strain, and another is rough or R strain.